All right, so welcome everyone. Thank you for logging on and attending this evening's program. My name is Matt Schumann. I am on the programming team here at Cary Library. Before we begin, just a few things to note. Please let me know in the chat if there are any technical issues that I can try to resolve. This program is being recorded and will be on the library's YouTube channel in a few days. And I will also send out a recap email with a link to that a few days after the program. If you have any questions or comments for our speaker tonight, please send them via the Q&A button. And this program is made possible by the generous donors to the Cary Library Foundation, as well as Lexington Living Landscapes. Tonight is part of an ongoing series of presentations where the library is partnering with Lexington Living Landscapes to bring experts in landscape and conservation issues to the public. So now please welcome Charlie Wyman.
Hi, and good evening, and welcome. We're delighted to have you with us this evening. Before I turn the floor over to Chris, let me tell you a little bit about Lexington Living Landscapes, which is, as Matt said, hosting this evening's program along with our friends at Cary Library. Lexington Living Landscapes was launched in 2020 for the purpose of encouraging more sustainable landscaping practices in town. So more native plants, fewer invasives, fewer chemicals, more trees. We were created as a partnership among 4 organizations, the town's Sustainable Lexington Committee and 3 nonprofits, the Lexington Field and Garden Club, the Lexington Climate Action Network, and Citizens for Lexington Conservation. We're here to help you create healthier, more nature-friendly yards and neighborhoods. To plug into what we do, go to our website, www.lexingtonlivinglandscapes.org. And there you'll find a host of resources, including news of upcoming events, how to sign up for our newsletter, and more. Our great thanks to Chris for joining us this evening and sharing his wisdom with us. Our thanks to Matt and Cary Library for co-hosting this evening's program, and to the Lexington Conservation Division for co-sponsoring this year's programs. After Chris talks, as Matt explained, we'll have a Q&A session moderated by another member of our Living Landscapes team, Sarah Bothwell-Allen. If you have questions, again, as Matt said, please put them in the Q&A and we'll get to as many of them as we can. Also, we have another treat for you. Chris has kindly agreed to lead a Firefly Walk at Willard's Woods here in Lexington next Tuesday evening, July 5th from 8:30 to 9:30 with the rain date of July 6th. The details are still being worked out, but we'll post them on our website in a couple of days, and Matt will include them in the follow-up email you'll all receive later this week. So let's get started. You are in for a treat this evening. Dr. Christopher Cratsley began researching fireflies as a doctoral student at Tufts University and has continued this research for over 20 years as a professor of biology at Fitchburg State University. Dr. Cratsley studies firefly flash signals and the interaction of fireflies and potential predators. He also collaborates with the Massachusetts Audubon Society on a community science project, Firefly Watch, focused on monitoring firefly populations in North America. Both through Firefly Watch and as a science educator, Dr. Cratsley uses fireflies as an ambassador insect to educate the public about the natural world. Chris, we're excited to have you with us this evening. The floor is yours.
Thank you very much. And thank you everybody for joining us here this evening. I'm looking forward to sharing what is, probably just a little bit of the many, many fascinating things that we could talk about with fireflies. It's always a challenge to me to figure out how to narrow it down in a way that engages people, but covers what's most meaningful and important. I hope I've done a good job of it, but the good news is we'll have question and answer time. So whatever I don't cover that fascinates you, for instance, things like how do we get more of them in our backyard? I'll try to mention that briefly, but I'm sure we can have a rich discussion of some of those questions at the end once people have had a chance to hear a little bit more about what fireflies are, why I find them interesting, and I hope that you will too. So I'm going to share a PowerPoint with you now, and just bear with me for a moment while I do that. And I'm going to present it. Okay, so hopefully everyone can see this, and we're going to go through fireflies from Lexington and beyond. And in fact, we do have a firefly here, one image of a firefly in the palm of my hand, not a Lexington firefly, but close enough, a Lincoln firefly. I did most of my big chunk of my doctoral work was done at a field site in Lincoln, Mass. So right next door. And in fact, if you look in my backyard here, if we see any fireflies, those will be Concord fireflies. I'm right here in Concord now. But fireflies are found around the world, on every continent except for Antarctica. And so the firefly on it's my right-hand side as I look at it, hope yours too, represents one of those from beyond, because I have had opportunities to study fireflies in other parts of the world. And so this is a firefly in Thailand, one of a fascinating group of species of fireflies that all flash synchronously. The males all flash synchronously, lighting up a tree. And so one of the things I draw people's attention to in this first slide is just that fireflies exist in over 2,000 species. And again, on these many different continents. But the basic plan for a firefly is fairly similar from one place to the next. So as far away as Thailand, as nearby as Lincoln, you'll see maybe some slight differences in color here, but the basic structure of the firefly is similar. And one of the fascinating things about them, one of the many fascinating things, is that so much of their evolution is around these bioluminescent flashes they produce, which is wonderful because that's both the magic, so as I say, their magic, and it's also their mystery. It's also what interests us about, at least in part, what fascinates us about them as scientists is how have they evolved into these many different forms of flash behavior, from these amazing mangrove trees that light up with synchronous fireflies in Southeast Asia and a variety of places, to, as I hope you'll learn to appreciate today, our local fireflies that, while most of the time not synchronous, are doing some pretty amazing things as well. So, and then finally, I'm going to touch a little bit on what we're doing to think about how we can conserve them. So here's a quick outline of what we'll be talking about today. The mysteries and magic of fireflies. I'll try to touch on a few fun facts about fireflies. Again, I think the fun facts will continue to be peppered throughout the talk because they just can't help but come up as we learn more and more about them. But I will refer you to some places to try to learn more about them, because again, I can't cover everything. Also, we will then delve into really the local firefly, identifying the groups of fireflies, what we call firefly genera, that we will find here in this area. And so as an organization like this one that's focused on how we can preserve our local habitats, we'll talk about three big groups of local fireflies: Photinus, Photuris, and Pyractomena, and their flash characteristics. And finally, I will spend a little bit of time talking about a wonderful partnership that firefly researchers like myself have with the Massachusetts Audubon Society for Firefly Watch, a community science project that enlists folks like yourselves to help us monitor firefly populations. And there's a couple different levels of involvement: Firefly Watch and Firefly Watch Pro, that I'll be introducing at the end of the talk. So again, if as any questions come up, please do put them into the chat, and I will jump right in with what fireflies are. This to me is sort of the first fun fact. I've already tipped my hand a little bit that there are, in fact, fireflies are not one thing. They are over 2,000 species worldwide, and as many as 200 species in North America. They are, of course, all animals, what we think of as multicellular organisms that get their energy by eating things. And so, like ourselves and the many other kinds of animals that we're familiar with, that's the kingdom that fireflies fit into. And if anybody remembers their high school or middle school biology and the process of these different smaller and smaller groupings into which we place organisms. You may or may not remember that the next smaller group we place organisms into are the different phyla or individual phylum. And so they belong to the phylum Arthropoda, or we can just call them arthropods, animals that have jointed exoskeletons like crustaceans, insects, and arachnids. And within that, they belong to the class hexapods, which is our fancy word for insects, arthropods with 6 legs and usually with wings, bees, flies, etc. And then this is really the key piece of information here to know, because you probably had figured out they were insects, but to understand that they are, in spite of their name, in spite of being called fireflies or sometimes being called lightning bugs, colloquially, they are actually coleoptera. They are beetles. And the main characteristic, or one of the key characteristics that we use to identify the beetles, are that they have these stiff forewings. If anybody can sort of envision a ladybug in flight, another beetle, we know that one of their 2 pairs of wings, most insects have 2 pairs of wings, the forewings, that pair of wings is modified as a hard covering, or at least a harder covering. And so in the case of fireflies, if you see them flying, you will see these forewings held aloft while the hind wings do all the flapping. And the hind wings are very thin and diaphanous and very similar to, again, what you'd see in a ladybug. So that's the beginning of what fireflies are. And I've put this really big family tree of the coleoptera up here, not because I expect you to be able to read it in any way, but to kind of remind all of us that the coleoptera are an incredibly diverse group. So of the land animals, the insects are by and far the biggest group of land animals, and of the insects, the coleoptera are far and away the biggest group of insects, over 350,000 species. So when I talk about the 2,000 species of fireflies, that's just one small fraction of all of these beetles. And the other reason I bring this up is to say, again, what are fireflies? So we know they're beetles. What makes them special amongst beetles? Well, one of the things that makes them relatively special, although not completely unique, is that they have bioluminescent larvae. That means that, like many insects, they undergo a complete metamorphosis from a larval stage to an adult stage. And we're really, most of us, most familiar with the adult stage that flies around for a couple months in the summer. Probably any individual species, maybe only 1 month, but if we were in the right place at the right time, we might see them throughout some parts of June and July, usually around here, not in August. But they are relatively unique in that they have these larvae bioluminesce. Now we know the adults also, of the ones that we pay the most attention to, bioluminescent as well, but there's more to that story. So the flashing firefly that we see, as we'll talk about in a moment, is not the only kind of firefly, but essentially all of them have larvae that we might call a glowworm. They glow. And they are related to some other insects and some other beetles that you might know from your garden. The closely related soldier beetles, Cantharidae, that are a relatively useful predator of some other plant-eating insects in your garden, and the click beetles, the Elateridae, that although some of them can be predators, also some of them can be pests on some of your plants. And this divergence occurred back in the Cretaceous period, the last age of the dinosaurs. And in fact, the Lampyridae, you may or not be able to make this out. They're at the very bottom here of this condensed version of the image I showed before. So it doesn't have all the beetle families. And then you can see some different Elateridae and Cantharidae up there. And I zoomed in just a little bit more on it to point out that there are some other species with bioluminescent larvae, the Phengodidae and the Rhagophthalmidae, and those evolved from fireflies in the late Cretaceous, a little bit more than 100 million years ago. And so those are their closest relatives. And some of these And in fact, even some Elateridae have adults that can produce bioluminescence like the fireflies do. But far and away, the fireflies are most characteristic of all of these groups in having bioluminescent adults, adults that can flash, adults that can glow, in addition to larvae that can glow. Virtually all of these ones, Phingodidae and Rhagophthalmidae, that's a mouthful, are able to bioluminescent as larvae, but the Lampyrid beetles are more unique in having so many representatives where the adults actually produce flashes, glows, flickers, a variety of forms of bioluminescence. And in addition to that, we actually know that their research by scientists have revealed that the bioluminescence in the Elateridae, the instances in which it happens, it's fairly rare, but the instances in which it's happened in those click beetles is in fact a case of parallel evolution. The two, although it's the same chemicals involved, those chemicals evolved separately and independently in the two different lineages. Whereas these groups, the Phingodidae, Rhagophthalmidae, and the Lampyridae, and that's our family of beetles that the fireflies belong to, all seem to have evolved this larval bioluminescence and the mechanisms for it from a common ancestor, pictured right back here in that late Cretaceous. Okay, so that's one of the fun facts about fireflies. But the other really fun fact I want to highlight for today is the answer to why they flash, or I should have really said why they bioluminesce or flash, because here is an example of one of those firefly larvae that I was talking about. Clearly, they look very different than the adults. Here's an example of an adult. And they are similar to the adult in one really important way in a number of firefly species that we've studied. And that is represented by this diagram here that shows the chemical representation of a group of chemical compounds that we call lucibuphigins. Lucibuphigins are chemical compounds that can be toxic to potential predators, and they provide chemical defenses to larvae of many firefly species and adults of many firefly species. And they happen to block a chemical pathway in many, many animals that's called the sodium-potassium pump. And they block that activity, and that can actually be lethal for some animals. There's cases where people have had pet lizards die from eating too many fireflies. And certainly it tastes bad. So anybody who has tasted a firefly knows that they taste bad. And we think that ultimately that ancestral evolution of bioluminescence in fireflies that happened in the larvae was as a warning signal to predators, warning predators that they taste bad. So the glowworms glow as a signal, similar to, we call these signals aposematic signals. And it's similar to the bright colors that you might see in a poisonous snake or poison tree frog that are, in that case, a visual signal that predators can see in sunlight. But in this case, fireflies and their relatives have evolved a visual signal that can be seen even in the absence of sunlight as a way to signal to a potential predator that they are chemically defended. And in fact, it makes a lot of sense because these larvae, in many cases, certainly the larvae around here of our firefly species in New England, are for the most part living underground, living in the soil. And another reason we may not see them as often unless we were to go out on a night like tonight where there might be some rain and they might have come up out of the soil and you might see them glowing by the side of a path there. But otherwise they're certainly not as easy to see as the adults, both because they're in the soil, they're small, and because they're giving off these faint glows, especially when they're disturbed. And that's one of the other reasons sometimes a rainy day is a good day to see them, because the drops of rain can be enough to disturb them and stimulate them to glow. So that's where we think bioluminescence first evolved in the firefly ancestors. But of course, that's not the whole story of why fireflies flash, because the flash in adults certainly may serve in part a function of warning predators. But as I've noted before, fireflies come in a bunch of different versions, some of which flash. And in fact, this is color coded. This is a figure from a 2008 paper by myself and my advisor, Sarah Lewis. And this paper shows in green species of fireflies. And again, this is not all the species of fireflies, just some representative ones that have bioluminescent flashes of light. Species that have glows, they just glow as adults. So again, these are now we're talking about the adults. So there are species where the adults just glow. And even species that don't have any bioluminescence whatsoever as adults. They're non-luminescent as adults. And so that's to point out that the evolution has occurred in a variety of ways. And the adult signal in particular has evolved in different directions because it's functioning not just as a warning to potential predators. And we have some evidence to believe that it is still at least secondarily can function to warn predators. We did some research on bats and fireflies that showed that you never find fireflies in the, well, to put it politely, the bat poop. I guess more guano is the more polite term, but you never find fireflies in having been consumed by bats and digested by them, and those hard, indigestible exoskeleton bits ending up in their feces. So we know they're somehow avoiding fireflies. We know that, in fact, they hate the taste of fireflies from our experiments. And finally, we know that when we fly bats in enclosures and use lures, they will attack those lures. This is a lure spinning on a very thin thread that's being powered by a printer motor. In our case, the bats would swoop in and try to attack those lures. But if we turned on a flash on those lures, they would avoid them. So there's still some evidence that even a flying and flashing firefly is protected in part from a potential predator by bioluminescence. But we also know that most critically, these signals that they're using, and whether it's a glow or a flash, and as we'll see, whether it's one of the many kinds of flashes that we can see around here in Massachusetts and across North America, in each of those cases, it's also being used as a sexual signal, a signal where males are signaling to females, or in some cases, females signaling to males, and vice versa. So that's really what we're going to key in on in terms of why do fireflies flash. Now, I've mentioned that there are some differences in the flash patterns. I'll just briefly highlight a few interesting examples. I've already mentioned the Southeast Asian fireflies that belong to a genus called Pteroptyx. And within that genus, a number of species, the males are sitting in a mangrove tree, all flashing synchronously, and females are flying into that tree to mate with males. Now that's very different, a very different sexual signal than the one we see in the most common European firefly. The most common European fireflies, the males are completely unable to flash. The males fly around with no lantern to flash, but it's the females that remain perched down in the vegetation. In fact, the females can't fly. They have a form much more like the larvae that I showed you, and they are glowing. So flying males in Europe from England, down into Spain and Portugal, there are some flashing species, especially as you get down into areas like France and Spain. England is pretty much isolated to just this one non-flashing species, but across Europe, this is the most common one, Lampyris noctiluca. Lampyris noctiluca fireflies, again, the females are glowing to attract males. And finally, I would argue that we have one of the most fascinating systems, but they each have their merits. Certainly, it's spectacular to watch the bioluminescent, synchronous flashes of the Southeast Asian fireflies. But we have a really wonderful system of sexual communication in these fireflies here in North America, and I'll be highlighting more of the details of it as this talk goes on. But in our case, you have males. What you're seeing out there in a field are males spontaneously flashing. The females, like in Lampyris noctiluca, are perched down in the vegetation. In some species in North America, the females can't fly, and other ones they can. And most of the ones around here, the females are able to fly, but they seldom do. They spend most of their time perched in the vegetation. But unlike those Lampyris noctiluca that are just kind of constantly glowing to try to attract males, females here of the species that we're going to talk about are waiting to see those male flashes and then responding with a flash of their own at a species-specific time delay after the male flash. So there's this wonderful courtship dialogue of male flashes and female responses that continues until the male finds the female. The male will perhaps land in the vegetation, and then you'll be able to watch him flash again, and then watch the female respond again, and he'll move a little closer and flash again. And you can see that go on, especially on a cool night where it may be harder for him to navigate to the female because he's moving more slowly. Whereas on a warm night, he might get in there really quickly, and you find the whole courtship is over very fast. But this courtship dialogue is relatively unique to fireflies across North and South America. So not so unique, a pretty big group, but we do not see these precise patterns as much in other fireflies around the world. So a wonderful thing about our fireflies that you can see right here. If you want to learn more, this is going to be my first plug for this wonderful partnership with Mass Audubon. Mass Audubon has the Firefly Watch Community Science Project, and this project has all kinds of resources for more information. As we talk at the end of this, I can give you other resources. There's a wonderful international organization that I happen to be serving on, fireflyers.org, which has its own website with some additional resources. So, but there's plenty of places to find more information and about these fascinating organisms. So, but as we continue on, let's focus in on the local fireflies that we have around here. And we identified that we have Kingdom Animalia, phylum Arthropoda, class Hexapods, order Coleoptera, family Lampyridae. And again, going back to middle school, high school, or even college science, we know that the next two groups, that smaller and smaller groups into which we place organisms, are the genus, or the plural of that are genera, and then finally down to the level of an individual species. So we're going to start by just figuring out what are three of the most common genera within the family Lampyridae that we can find here in our backyards in Lexington and really throughout the United States. Here they are, Photinus, Photuris, and Pyractomena. And again, you'll find more information about them on the Firefly Watch website. If you take the time to catch a firefly in your backyard here or in a park or anywhere around, you can often get a pretty good sense of which of these three genera you're looking at by some physical characteristics that they have. We're going to find that that's not so possible when we get down to the level of species, but for the level of genus, these three groups of closely related species can be distinguished by some basic patterns. So I'm going to introduce you to a little bit of terminology that's common across the beetles. I mentioned these hardened forewings. They are called elytra. And then beetles also tend to have this, what we'll call a head shield, but it actually has, again, a scientific name, a pronotum. So the color patterns on the elytra and the pronotum can give you a clue as to what you're looking at. So the Photinus and Photuris fireflies, well, actually all three of them have these pale margins around, pale yellowish margins around a dark central part of the elytra. Now you'll notice Photuris, not always, but in some cases, has a distinct stripe as well down the elytra. So that's one way. If you see, catch one of these fireflies, take a picture of it, look at it, and it has a stripe down the elytra like that, then you can pretty safely say you're looking at a Photuris. Unfortunately, if it doesn't have that stripe, you can't absolutely say that it is not a Photuris, because there are some Photuris that lack that stripe. But it's a good way, if it's there, good way to know it's a Photuris. The other big key to telling it's a Photuris, while I didn't make these specifically to scale, I did represent the Photuris as bigger because they are a larger group of fireflies. And this is where we start to layer in some of the fun facts about fireflies, even to this part of the talk, which is as you look at the legs of the Photuris, you can somewhat see in this image, but it's really very apparent when you start to look at these things side by side that its legs stick out much wider from its body than these other two groups of genera. And that's because, so you add the large size and these wide arms together, the Photuris are actually capturing and eating fireflies from these two other genera. So these are predatory Photuris. In particular, the females are highly predatory on Photinus and Pyractomena fireflies, and particularly on males of Photinus and Pyractomena fireflies. In fact, they were dubbed by the person who discovered this behavior femme fatales, because they will in fact simulate the flash responses of females of Photinus species or Pyractomena species in order to capture and eat the males. And if you're a firefly researcher out in the field looking for female fireflies, which often involves going out with a pen light and pretending you're a male to try to trick the females by producing characteristic flashes of the species that you see out there, you'll in fact find that, yes, the females of the species you're trying to find will respond, but sometimes you'll be fooled just like a male, and you'll find one of these large Photuris waiting for you instead of a Photinus or Pyractomena firefly. So other characteristics to tell them apart. I mentioned the pronotum. In general, especially around here, Photuris and Photinus have relatively similar pronotum in that it has the yellowish margin, black stripe down the middle, red dots on either side, or pinkish dots on either side. The Pyractomena, many, but again, not all, have instead a dark margin on the outside. So again, if you see a dark margin on the outside of the pronotum of the Pyractomena, you can fairly safely, or of the firefly, you can fairly safely say you're looking at a Pyractomena. Again, these are the bioluminescent flashing fireflies that are out there. And the other thing that's a little bit more characteristic is that the pronotum, in terms of that, pretty much every Pyractomena has this, is that the pronotum, you can kind of see it has a ridge to it. It comes more to a peak than you see with the more flattened pronotum of Photinus and Photuris. And similarly, in terms of the way the elytra meet together, you'll see more of a peak and a hunchback to the way the elytra come together in Photuris when you compare it to Photinus and Pyractomena, where they're more flat. So those are a few clues to try to tell what you're seeing in your backyard. And a quick summary of them: these three genera of land-period beetles you're most likely to encounter around here: Photinus, Photuris, and Pyractomena. Those darkened lateral margins, if you see them, then you know you're looking at a Pyractomena. Although if you don't see them, you can't completely rule out Pyractomena. And those pale stripes, same thing for Photuris, although the long legs and larger size are also really helpful. Okay, now if you can find a female using the techniques I described, that's another great way to tell these three groups apart. The males you can't really tell apart by their lanterns. The lanterns of males are pictured here. Here is, again, that story of the Photuris female devouring the Photinus male, and you get a better sense of the difference in size that can exist between species in these two different genera. But whether it is a Photuris or a Photinus or a Pyractomena, the males have the lanterns within these two segments. So here's Pyractomena, both segments. Here's Photinus, both segments. Where we can tell them apart are the lanterns of the females. Female Photinus lanterns just occupy the center of one segment. Photuris female lanterns, you have to look more closely because they look almost like a male lantern in that they have the translucent material makes up both of these segments, but the actual lantern itself is constrained to a narrower region of that than the whole lantern like you would see otherwise. And finally, in Pyractomena females, they're on the lateral margin. So a couple other things to look for when you're out there. Males and females of each genus have these different lanterns. The male lanterns are almost identical across the genera, but the female lanterns vary with those narrow bands on a single segment in Photinus, wider bands across two segments in Photuris, and those two lateral bands on the two segments in Pyractomena. Now, it is hard to find females. I mentioned they engage in this courtship dialogue with males. They're observing a male flash and responding to it. And so if you're just going to try to find them that way, find them when they're responding to males, you may have a hard time because the male may find them before you do. And once they go dark because they're mating, you're not going to see them. That's why I say you may want to go out with a pen light and try to simulate the flashes of the male, especially maybe late at night. After the males have stopped flying, there may be some females that didn't succeed in mating, especially as it gets to be late in the season, and those might be still there responding, and then you can find them. But they are harder to find because they're down in the vegetation. It's very easy to disturb them and have them just stop responding whatsoever, and it is also quite, their flashes are much dimmer than the male flashes in many cases. So. But it's a fun activity. I encourage it strongly. Go out there when you're looking, add this to, we all go out there and watch the males flying around, but try to see if you can see the females as well. Okay, now back to the males, although we're going to learn about, again, the female response, certainly back to this courtship dialogue in general. Once we get down to individual species, there's this wonderful, fascinating diversity in the ways in which they flash. So this is just a sampling of species that belong to the genus Photinus. And it's representing the male flash patterns and the way they differ from species to species, with males producing a single pulse repeated at regular intervals. But those intervals in one species might be about 3 seconds, in another about 4, in another 5 or 6. So really interesting differences that occur in the way they produce those single pulses. And some of them will produce not single pulses, but pairs of pulses one right after another that are then repeated at a regular interval. And so here we have 3 different species that are producing double pulses. And finally, some of them will produce trains of multiple pulses. And those might even be variable, where it might be 3, sometimes 4, sometimes. And so we can see examples of those as well, where they'll repeat those at regular intervals. So just within Photinus, an incredible diversity of flashes, a diversity that helps us tell them apart. And it's not just in the male flashes, it's also in what we call the female response delay. This right-hand column is showing the time from when the male produced a flash to when the female responded. In some species, the females respond very, very quickly, within a half second of the male producing the flash. In others, they wait 3 seconds, 4 seconds before they produce their response flash. And again, sometimes when the males produce multiple pulses within their flash, the females will respond with multiple pulses within there. So fascinating diversity of flash pattern. And this is, in many ways, the only way that we can tell individual species apart within a genus. It's not the only way. We can catch them and actually dissect them open and carefully look at their genitalia to tell them apart. But it's certainly the way that in a quick way, the general public, anybody can go out and try to get a sense of what they're looking at out there in their backyard is by trying to observe these flash patterns and this courtship dialogue of what is the flash pattern within the males and what is the response delay within the females. Now, the other thing that sometimes gives you a clue is that many of the Photinus flashes are relatively yellowish in color. And as we'll see, Photinus have this diversity of single, double, and trains of multiple pulses, whereas some other species don't have as, well, they have diversity, but in different ways. So the other thing I'll point out is that this is Photinus pyralis, a species that we don't have around here, but I highlight it to make one more point about them, is that sometimes there's also a very characteristic way of firefly species will fly as it flashes. And pyralis is famous for producing these J-shaped flashes, flying upward and forward as it flashes, and produces that long duration flash. And so that's another thing. And other ones that we have around here, you might see them producing flashes like Marginellus. Doesn't produce a J shape, but as they produce those flashes, they often are flying upward as they produce them. So some things to look for. As we move to another genus of fireflies, Pyractomena, you're going to notice that they don't have examples, that especially around here, we don't necessarily see examples of Pyractomena that produce multiple pulses or trains of pulses. These ones are producing more of a flickering pulse. You can see it's not multiple discrete pulses, but that's meant to be a flickering pulse. In this case, we put the female response delay, or not we, but the person who created this put the female response delay right next to the male flash. But you can see that Pyractomena produce single pulses repeated at regular intervals. Sometimes those pulses are very long in duration, and sometimes those pulses have a flicker to them. And again, there's different female response delays. Also, some Pyractomena, and this one in particular that we have around here, Pyractomena borealis, produce an amber flash. Their flash is much more orangish in color, and that's a way that you can tell them apart. And finally, we have the Photuris. And Photuris have an equally diverse set of flash patterns to Photinus, in fact, arguably more diverse, because they have these some fascinating cases where their flash actually ramps up and becomes brighter and brighter, or dims down over the course of the flash. And again, they have trains of multiple pulses. This is something you'll see a lot around here, Photuris versicolor, that will produce these trains of multiple pulses. And often those pulses may look more green in color to you. Again, not true of every Photuris species, but just something to keep your eye out for. Are the flashes you're seeing more amber, more yellow, more green? Worth looking for and giving you a sense of what you might be looking at. Okay, so that's how flash patterns vary between species, and with that, those broad and not definitive patterns, but things to look for of yellow flashes, amber flashes, and greenish flashes. But really what you want to do, if you're fascinated by this like I am, is you want to go out, catch them, figure out which genus you're looking at based on the physical characteristics that we discussed earlier, and then jump in and see what are you looking at in terms of within that genus, which of these species it is. And that's where we go back to really trying to count what you're seeing out there. I always do it in my head, and you have to be sensitive to the fact that you could try it with a stopwatch tube. You have to be sensitive to the fact that this is a temperature dependent thing. As it gets colder, all of the time elements of a particular species will become longer. As it gets warmer, they will become shorter. But it gives you a general sense if you watch a single pulse and then count in your head the number of seconds, 1 Mississippi, 2 Mississippi, 3 Mississippi, until you see the next pulse by what you think is that same firefly. Again, part of the challenge, if it's chaotic out there with a lot of different fireflies, you get a sense of what we call the flash pattern interval, that time from one pulse in a pattern to another. Now, we use that same term flash pattern interval, even if you're using it a double pulse species, timed from the first flash of a pair of pulses to the first flash of the second. We call the time between those pulses the between pulse interval. So, a double pulse species is going to have two different things you're going to want to count. You're going to see a flash, count 1 Mississippi, 2 Mississippi, and then it might flash again. And then you say, okay, that's 2, but then you keep going, 3 Mississippi, 4 Mississippi, 5 Mississippi, until it produces the first of the second set of pulses. Once again, similarly, if you're looking for those female response delays, whether it's to your pen light or to a male flying around flashing, you're going to try to count how long is it till, how many seconds is it till you see that female produce the response. And then you can also kind of get a sense of how long in duration was the pulse. If we go back again, you can see that certain species have durations that are on the order of a couple of seconds. Although in general, many species that we're looking at and there's another one with a multiple second duration, potentially even longer. But certainly in many cases, we're also just saying, well, is it really, really fast, much less than a second, or is it close to a second? That might be something that you could get a sense of for both the male flash and the female response as well. See some very, very quick female responses, some longer duration female responses. So that's what's fascinating. And going back to the fun facts about these fireflies, is that when we have a situation like this, where you see this diversity of flash patterns within a genus, or as we say, a diversity of sexual signals within a genus, without much diversification of the rest of the morphology, the rest of the characteristics, the rest of the body plan of the fireflies. We can't tell these species apart within a genus just by looking at what they look like. That suggests to us that a process called sexual selection, a sort of special form of natural selection, has driven this diversification of species, where in some ways individuals are, new species are emerging because individuals within a population are experiencing some advantage in terms of their reproductive success. So not their survival per se, but their reproductive success and their ability to pass on their genes because of some characteristic of their flash pattern. And this is what I did my thesis work on many years ago, determining that, in fact, females of a particular species, a couple different species of single pulse fireflies, the Photinus pyralis that I showed you that's not around here, that I studied down in New Jersey, but also Photinus ignitus around here in Massachusetts and Lincoln, that females are preferring males with longer flash durations than the average duration of a male flash. Other people have found things like when there's pairs of pulses, that females seem to prefer the pairs that are produced more quickly relative to each other. When there's a train of multiple pulses, again, when those pulses come in at as fast a rate as possible, there seems to be female preferences. But there's also a limit to it. When I looked at the female preferences for male flash duration, yes, they preferred slightly longer duration flashes than the average, longer duration. But if the duration became too long, outside of the range of what they would ever see in the species, then they weren't responsive. So males getting some advantage, potentially based on the characteristics of their flashes, and that potentially driving the evolution of different characteristics of their flash patterns. So we're going to do one last exercise with flashes, which I think is kind of a fun one. And that is just to show you some animated examples. So they're not actual fireflies, because it's pretty hard to get good videos of actual fireflies doing these flash patterns. But cartoon animations of them made by the folks at Firefly Watch. And I take responsibility for the error in this first one. I mentioned to you that there's an amber flash quite often in Pyractomena, particularly Pyractomena borealis, which we've animated here. Somehow, I think when I saved the file from maybe PowerPoint to a Google slide, something like that, the color changed on me. So it's not going to look amber, but let's look at an example. And the Pyractomena borealis fly in quite often on the margins of tree-lined margins. And so that's what we've tried to represent here in our animation. And you can imagine yourself now out there looking at Pyractomena borealis. Granted, I think it's going to look purple instead of amber, but and counting in your head when you see a flash. There's 1 Mississippi, 2 Mississippi, 3 Mississippi, 1 Mississippi, 2 Mississippi, 3 Mississippi, 1 Mississippi. So we're seeing about a 3-second interval. And again, we're only seeing single pulses. And as we mentioned, Pyractomena males tend to produce single pulses or flickering flashes. And you could, going back to our chart for a moment, you could say, okay, 2.5 to 3 second interval or flash pattern interval. And that's pretty characteristic of borealis, whereas lucifera, very different, more like 5 seconds you'd have between the flashes. Let's look at Photinus, and we're going to look at 2 examples of Photinus just because you are definitely going to see around here in Massachusetts, both single pulse species and double pulse species. So I mentioned I spent time in Lincoln studying Photinus ignitus. In the very same field where I studied those Photinus ignitus, other grad students were studying Photinus greeneye, which were producing pairs of pulses. And we'll start by looking at the Photinus ignitus. You can see it's got about a 5-second flash pattern interval. 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi. So I'm hoping these flashes are showing up on your computer screen. Often I do this in front of a live audience and we have the lights turned down, but if you're seeing them, you can imagine yourself, and this is of course more of the habitat of ignitus. They tend to be more out in fields than under trees. And so we've got that represented here. But that would be your way to see if you're possibly looking amongst these at Photinus ignitus, because it has one of the longest flash pattern intervals, certainly of what we see around here. Pyralis, I mentioned, it also has a very long flash pattern interval up to about 5 seconds, but Pyralis is not found in Massachusetts. I had to go down to New Jersey to study them. So they are found, Pyralis is found all over the place. And of course, you could also tell Pyralis apart. I mentioned it has that interesting J-shaped flash. If you ever are observing fireflies in another part of the country, you're very, very likely to encounter Pyralis if you're outside of New England, because they are widespread all the way out to Texas and New Mexico and across the Midwest and down into Florida. So they're everywhere. Okay, let's look at Photinus males producing single, double, or multiple pulse flashes. And let's see, yeah, that was just the single pulse Photinus. We did that already. Okay. And then finally, well, let's look at a Photuris. So here's a Photuris again, Photuris versicolor, you might see around here, producing those trains of multiple pulses. 1, 2, 3, 4, 1 Mississippi. Well, you want to count from the beginning. So 1, 2, 3, 4, 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5 Mississippi, and we may not have another one there because it flew over the hillside there. But they're also they can be very fast flying too. So it gives you an idea. Wide range of flashes in that species sometimes, and again, sometimes they will mimic Photinus. All right, well, the last thing I wanted to talk to you about today, after introducing you to the fireflies that you're going to see around here, is what we're doing to try to protect them. And this takes us back to this citizen science project. Now, what you want to do is identify a firefly habitat to study, even your backyard, and you can register online at Firefly Watch and describe your habitat. Now, by the way, this is an interesting example of a daytime firefly, and here's another one. We do have around here, and let me just actually go back for a second. We have around here fireflies called Elychnia corusca. The adult males and females have no lanterns, but they are lamp-eared beetles. The larvae are bioluminescent, and they're actually out in the wintertime as well. And Lucidota is another example, another genus with a couple different species of daytime fireflies. And these are actually even found out on the West Coast, places like California that have no flashing nocturnal fireflies. So actually, sorry, I got talking about that, but when in your chosen habitat, note the date, time, and weather data, and we ask you for 3 10-second intervals to count the number of flashes seen. Now, why are we asking people to do this? Well, the bottom line is that we have heard time and again, those of us who do research on fireflies, that people feel like they don't see them the way they used to when they were kids. And there's a couple possible explanations for that. We know that invertebrates and vertebrates and many species of all kinds are disappearing across our planet. That's been really well documented for many species. It has not been well documented for fireflies. In fact, we just in 2021 concluded the first US and North American assessment of firefly species. And this is done in collaboration with an organization called the International Union for the Conservation of Nature. And what IUCN does is it has a standardized process that can be used across the world. Now, we have a process in the United States that allows us to place a species on an endangered species list. This is analogous to that, but the process is something that can be used on any species in any country. And in fact, doesn't just say it's endangered or not endangered, but identifies different levels of threat. And so there were no fireflies in Massachusetts, you'll be glad to hear, that were identified as at risk of extinction. But there were some in the US that we consider of concern at different levels of concern. And the other thing was that we found that 40% of the species that we wanted to report on, we had insufficient data on. We could not report whether they were at risk or not. There just wasn't enough known about what their populations were like. And so that's a big part of what Firefly Watch is doing. Now, we've been doing it long before the IUC, we did this IUCN Red List process because, again, we were just interested in seeing overall were firefly populations declining. And once again, our data doesn't suggest, and that's maybe not even just because of the Northeastern bias, but also because we did have data from other, people do put in data from other states into this Firefly Watch system. I think it has more to do with over the 10 plus years we've been doing this, we have been just asking people, to tell us how many, over these 3 10-second intervals, how many flashes they see. And that is irrespective of species. And some species, as our red list suggested, are doing just fine, and some species are more at risk. And so what we've been doing over the last few years is to encourage people to start with Firefly Watch, observing 1 time a week, again, providing that information about your location, your habitat. These are a number of the things that we think impact firefly populations, how frequently an area is mowed, again, what the weather is in your area, whether there's sources of artificial light that might impact firefly populations. And then once again, count that total number of flashes 3 times, 10 seconds each, and also tell us a little bit about how many different flash patterns did you see out there? Did you see just one? Did you see a couple different types of patterns out there? And it gave us a very general sense of, are there fireflies at sites, and are there a lot of fireflies at sites or few fireflies at sites? But we're now encouraging people, once they've tried out Firefly Watch, or even in some cases if they haven't and are just really gung-ho, to try Firefly Watch Pro, where we move to 2 times a week observations, providing some of the same information, a little bit more info about pesticide use, whether you have fire ants, other disturbances, but actually getting down to trying to identify a single kind of flash pattern, so that you feel like you're looking at one particular species, and try to tell us what that flash pattern is, and we'll work with you. And of course, tell us also the genus you think it is based on the identification clues I described to you, to see if you can figure out what species you have in your yard or your park nearby or fields nearby, whatever it might be, so that we can begin to ask the question, for an individual species of fireflies, what's happening, and in particular, what might be happening over the next 5 years before we're going to try to do our next red list assessment of fireflies. So Firefly Watch Pro, you log your data on a computer like this. We don't need to go into the details of it now, but if you are more interested in this, again, the Firefly Watch website can put you in touch with me and others who can help get you started on this work. And we really welcome you getting involved and telling us all about what you see. So I'm not going to dwell on all of that stuff. I'm going to just jump ahead now to wrap things up and really allow some time for questions. I'll say one last thing though. I did skip over this slide quickly. We also encourage people always, and this is a big initiative at Mass Audubon as well, not just for fireflies, but for birds and other animals, to load your observations. If you do catch a firefly, if you take a photo of it, put it into iNaturalist. It's a great way to have other people help you figure out what genus you might be looking at. Again, very hard to necessarily know the species without the flash data, but at least get you started on knowing what you might have that you're looking at in your backyard or nearby. Okay, now questions.
Thanks very much, Chris. That was really interesting and a lot of detail. We've gotten a number of questions. I think this one that was just about specifically a detail of Firefly Watch first, because that was what you just ended on, which was for doing it 1 to 2 times a week. How many weeks is the season for doing that if you're participating?
So that's a really good question. And we don't dictate it for anyone because, of course, it depends on the fireflies. And it depends on where you're located. Around here, I encourage people to start looking in maybe the latter half of May. I was running a bunch of programs for Mass Audubon all in the latter half of May, and on only one of them did we see fireflies. We've had some really cool nights. We had some really cool nights, particularly in May this year. But then as June rolled around, obviously we've been seeing many more of them. So it's always good to look early. Obviously, you don't have to log any data until you start seeing them, and then you continue ideally until you stop seeing them. And pretty reliably around here, that is the beginning of August. They're almost always in Massachusetts. They're almost always gone by the beginning of August, and so you might be observing them for anywhere from 1 to 2 months, depending on when they start and when they end. So, and of course, I guess the one other thing I'll say is we're always happy for data. Even obviously, we understand people people have vacations, they have other plans. We encourage you to get involved and give us what data you can, because some information about what's going on is always better than no information.
Thanks. And then another person asked the question about if they're not in Massachusetts, if they're nearby in Connecticut, can you use their data too? Like, what's the geographic extent of the project?
Yeah, the geographic extent of the project, we tend to focus across the United States. Now, I would say that we've got a strong, Midwest to Southeast to Northeast bias. The reason for that is that I mentioned there's no fireflies in California, Portland, Oregon, none on the West Coast. There are fireflies in places like Colorado, Utah, New Mexico, Texas, but especially in Western Texas, and again in those other states I mentioned, they are very, very rare. So there's actually another project out there that is called the Western Firefly Project. And instead of asking people to, report back, count them, all those other things, they just say, please tell us if you see them, because, because they're in so few places. One of the things about fireflies is that they are very moisture dependent. Those larvae that live in the soil really do need moist soil. They're predators in the soil. They're predators on soft-bodied invertebrates, things like earthworms, other insect larvae, and snails in some cases. And as they, as predators of that kind, Their prey tend to also be very moisture dependent, and they as well are going to potentially dry out and die if the soil is not kept, if the soil is not naturally moist enough. We tend to find them even around here, closer to riverbanks and bodies of water, although you can find them on hillsides sometimes too.
Thanks. Actually, that leads us into, we've had a number of sort of habitat and ecological questions, both about, what you described some different types of like forest edge versus meadow. What would you characterize the general habitat of fireflies that we have here in the Northeast? You've mentioned the moisture. Obviously, they're not going to be in as much in a very arid area.
Yeah, so you find them both, in forested areas and in open fields. I've done a lot of my study of the ones in open fields. They're in some ways a little easier to study because you can move around in those fields more easily to study them. And also they are the ones that, at least to me, have some of the most spectacular displays, because you can have, them, thousands of them flying around a field completely lighting it up. I often find that the ones that live in the tree understories tend to not be at the same high densities of them, but they still can be beautiful as they fly through those habitats. So you'll see both of them, and as you alluded to, the moisture is one of the keys. Looking for those places where they're going to, although it's not, again, here in New England, it's not so much of an issue. There's many places that are going to have moisture. I think one of the key things is that we don't find them in manicured lawns around here. One of the reasons that I would point to try to, there's a couple of reasons, but to try to explain why the species that we don't have around here, Photinus pyralis, is so successful in other parts of the country, spreading, out into places. It's one of the species you find out even out in New Mexico that can be obviously very dry habitats. Is that they do seem to be able to survive in dry habitats, which has also meant that even in places that are otherwise moist, but where people do things like mow their lawns, which makes the soil much less moist, because obviously there's a lot more evaporation able to happen when you have closely cut grass as opposed to tall grasses and plants in your yard. Photinus pyralis, I've seen it surviving. I was collecting it in athletic fields and other places in New Jersey because just their larvae can really handle dry conditions, but larvae of species around here simply can't. And I've never seen them in manicured lawns around here for sure.
For the species that tend to live either at the forest edge or more into the forest, are those ones that if you had a habit of not removing all the leaf litter in the fall, that would help support them maybe in your own backyard a bit better?
Yes, absolutely. And I think, we can point again to their predatory lifestyle as larvae. Certainly it's going to, once again, be beneficial just directly for them in terms of not, the soil will be moister for them. But I think what's even more important is that their prey are more likely to survive. So in Connecticut, there was the Canaan Land Trust a few years ago had me go down and give a talk to them, and they actually have set aside a firefly habitat, a firefly sanctuary in New Canaan, and a gentleman there also in his own yard had created a firefly habitat, and he'd done it very much by not mowing the yard, obviously not using pesticides or herbicides, and allowing, and even in some cases adding and supplementing, rotting logs, leaf litter, things like that would provide an ample source of potential prey for the fireflies in the form of soft-bodied invertebrates that were living in and under and around those decaying organic materials. And you could see, it was shocking to watch his yard, which was also next to the Firefly Sanctuary, and then immediately across the street, he had a neighbor who he loved to point out had this beautiful manicured yard, and 20 feet away, no fireflies, his yard completely full of fireflies. So striking difference.
Right, one living and one not. Somebody asked a question about whether fireflies are helpful or harmful to vegetable gardens, and I assume that would be a bit more in the adult life stage. You were talking about the one genus whose females are predatory on the male of other genera. What do the other?
Excellent question, and something I should have mentioned. As I say, there's always so many facts. I try to pick a few fun facts, and I try to get people to know what they might be seeing in their backyard. But the truth is that for most adult fireflies, they don't feed as adults. They are relying entirely on their larval energy reserves. There may be some exceptions to that, especially as you get to populations that live in areas where their life cycle isn't as seasonal. But when you think about it, for many, many fireflies that are living in places like the Northeast, they are, just by their very behaviors, they're limited in when they can engage in reproduction. They can only engage in reproduction when it's warm enough at night for them, because they're a cold-blooded organism. You could have a night where it's in the middle of July, but it drops down to 50 for some reason, right? We have those nights every now and then, and you go out in the same field that was full of fireflies they're there, but they're just not active because they can't warm up, but their bodies don't warm up enough for them to be physically able to fly and flash effectively. And so with that limit of here's the time period when they can mate and reproduce, they spend all of their efforts just on that mating and reproducing, and none spending time during the day trying to get any food that would allow them to live longer.
So wow, so they're not even doing any nectar feeding for sugar?
No, again, I've heard some reports from places like Tennessee and Florida, and it was either Bermuda or the Bahamas, of fireflies that have been observed on some flowers, observed on goldenrod. We don't really see it around here. And even then, there's the question of how much of it is nutritional, and how much of it might even be, if they are doing it, nectar feeding for the purpose of moisture. We do know that moisture is certainly critical to them. And so they're going to be spending their time conserving their energy and conserving their moisture as well under leaves, down in the vegetation. That's what you see adult fireflies around here mostly doing.
Thanks. So it sounds like their biggest ecological impact is really in their larval stage, when they're in a leaf litter, predatory, and as you were talking about, you do research also on what's predatory on them. I was wondering if you could talk a little bit more about that, and particularly in light of the local species we have that doesn't flash, are they less distasteful?
So a couple different things there. First, it's a good opportunity for me to just make the point when you've just highlighted their role in the ecosystem, and it is not, it's not what we think of as an ecosystem service role, right? There are those organisms that we point to as critical for an ecosystem because they, in some ways, are functioning as some critical service to the ecosystem. And our top predators, generally, because that's what they are in this tiny little soil environment, they are a top predator. They're not, and especially because they're chemically defended too, they're not serving an ecosystem role in that place. What I point to them as is they are the canary in the coal mine. As a top predator, they're the thing that's letting us know if this habitat is supporting a rich, diverse set of organisms of the kinds that they're going to feed on, right? So if there is a rich soil environment full of soft-bodied invertebrates, then you're going to see fireflies. And fireflies are, in that way, for me, an indicator organism, as well as an ambassador organism for the rest of the invertebrates that maybe aren't as fun to watch always.
Thanks. I think that's a really helpful framing for people to think about. So I appreciate your saying it in that way. We have a number. Oh, go ahead.
Well, you did ask about their predators.
Oh, yeah.
And they're spiders do feed on them. And you can see ants sometimes feeding on their eggs. Most fireflies, again, even what we call the diurnal ones, the daytime ones, are chemically defended. What's one other really interesting fun firefly fact, though, is that the Photuris, those predatory Photuris, are not chemically defended by lucibuffigens. And in fact, that appears to be why they evolved the predatory behavior, or somehow in concert with not being chemically defended. We don't know, which came first, the loss of the chemical defense, because it does seem to be fairly ancestral to the related group of organisms. But they are in fact feeding on these other fireflies, in part at least, to get the chemical defenses, more so than to get the nutrition, but just to get these lucibuffigens, because we can then see that they provision their own eggs with lucibuffigens. So the eggs of those females are chemically defended, even though the female herself was not chemically defended before she ate a Photinus firefly, for instance. So that's another fun part of the story.
That is a really exciting story. I would love to talk more about, but I don't want to ignore kind of this other big question people are asking a lot about, which is related to light pollution and various habits people have. One person mentioned they have a neighbor who hates insects and lures them with a bug zapper, and another person who loves and misses fireflies, so she's installed little lights to imitate the good feelings she got. And then just generally, like how much do just point light light sources, or just sort of the ambient general light pollution we have of being like near highways and cities, like how do those different types of light pollution affect these species, and maybe differently for different species, I guess?
Yes, well, and the last point you made is an important one. We always have to, as scientists, add the caveat that it depends. And that's really important. I mentioned Photinus pyralis, this species that we see across so many places as being very resistant and be able to survive as larvae in what seem to be pretty dry habitats. It also happens to be a species that comes out and starts flashing at dusk. So it's flashing and active when there's still a fair amount of ambient light. And we see that for some species around here. I mentioned Photinus greeneye as a double flashing species. And Photinus greeneye is another example of a species that comes out a little earlier at dusk. And so we could imagine that those species are not necessarily as impacted by light pollution, although you could also imagine they're impacted in a different way. So we know there are definitely species that are very negatively impacted by light pollution, where the light pollution is simply going to disrupt their flash behavior. They're not going to emerge and exhibit the flash behavior. They're not going to start flying in areas where there's a lot of light pollution. They're going to have more trouble finding their mates. Their mates may, females may not be as responsive because the females are being inhibited by the light pollution. But even for species that are evening flyer, the early evening flyers, they may find their flight period extended, and that might have negative impact. So there's a lot of questions, and there's going to be a lot of variation. I will say that that pretty much every bit of research that I've seen about this has certainly shown that that it's a problem, that it's not good for the species that are being impacted by it, even though there is variation from species to species how it can affect them. There's been countless different studies that have shown a negative impact of light pollution on different firefly species. So one of the things, so we just had a conference on fireflies just a couple weeks ago, and there's a lot of questions people have about, are there different colors of light that are better or worse? Again, it's important to note the answer is it depends. As you saw, different species produce more greenish or more orangish flashes. And some people say, oh, well, red light might not be so bad, but red light is going to certainly be more impactful for a species that is producing and responding to orangish flashes. And some people say blue light might not be so bad, but blue light is going to be more impactful for a species that's producing and responding to greenish flashes. So that's something to highlight. And I was very struck in the conference as well. Someone did research. Now, again, this is not on North American fireflies. This was, I believe, on the European firefly, Lampyris noctiluca. But they found that in that particular species, as long as they kept a light interference under 15 minutes in length, that the female could return to her normal nocturnal behavior. But for durations longer than 15 minutes, the female would never recover for the night and would never, continue to exhibit the normal flash behavior that she would exhibit. And that was striking to me, just something to think about as we, some of us also choose to have some kind of motion sensitive lights on our homes, and to, certainly keep, if you do that, to keep the duration, the on time for that after it's motion stimulated to a minimum. So anyway, lots of interesting things to think about. I just sort of spewing different ideas here about
you're touching on things related to some specific questions people have asked as you've continued talking. And so maybe just narrow in a little bit on people are asking, like, both what's the best time of evening to see them, twilight or dusk, and other people are asking, like, if you have lights in your yard or at your house, like, is it okay if you turn it off by midnight, or should you really just have it off except for motion sensitive, so that you just have it on for the few minutes you need it when you drive in and out?
Yeah. So, once again, it's, when you're going to see them is going to depend on species. But if I were to highlight a peak firefly activity period for many of our local fireflies, and again, it's going to vary as the summer goes on and sunsets. We're now reaching the point where sunsets are coming earlier and earlier and earlier. But from about 8:30 to 10 p.m. for me has always been the prime activity period for many of these species of fireflies, particularly in the genus Photinus. The genus Photuris is a little bit more special because I had a friend, I was visiting out in Brimfield, Mass yesterday, and he says he sometimes has trouble sleeping and wakes up at like 2 in the morning and goes outside and sees fireflies out. And I said, you must be looking at Photuris, because you'll see flash activity from them much later into the night. But again, I think a peak time to look for them would be from, at this time of year, especially from around 8:30 to 10 or so.
Thanks. I think that's really helpful for people to hear. We have a lot of people who, would really like to do their part to help them succeed in their communication at night. And also, if you want to talk to a neighbor about reducing light pollution, it's really helpful to have that kind of specific information, so that you can help them decide, well, what can they do that's not a big imposition versus what they assume is standard operating behavior. We had a couple other really specific questions. One is, what's the best website or book for identifying species by flash in New England?
So the best website, in my humble opinion, is the Firefly Watch website. You will find the flash charts that I showed you online there. You'll find some other information as well. The best book, and I'm probably going to butcher the title here. I actually meant to carry it in here with me and hold it up, but now I'd be taking a little bit of a walk to another room to find it. But it's Fireflies, Lightning Bugs, and Glowworms. I think that's the correct name of it. The author is Lynn Faust, F-A-U-S-T. And it is really it is the authoritative field guide for Eastern fireflies. Lynn is based out of Tennessee, and there is, there's more of a focus on fireflies south of here, but it certainly includes the species that we have here. And so, yeah, that's what I would point people to. It's an excellent field guide. So much information of it. So Fireflies, Lightning Bugs, and Glowworms. I think I've got the right order there, or it might be, I know it starts with Fireflies, but again, Lynn Faust, excellent book. And then I also do always like to pitch another book that, if you're interested in the science that I've been talking about, as well as the beauty of it, Sarah Lewis, who had been my advisor at Tufts when I was there, and continues to be just a leading figure for firefly research, and now especially a champion for firefly conservation, really one of the integral people in getting the process of red listing fireflies across the world going and continuing. And that red list is the process I described before of the IUCN going species by species, really firefly specialists working with the IUCN to a species by species. So she wrote a book called Silent Sparks. And again, that's Sarah Lewis, Lewis. Excellent book that talks about so much of this science in detail, and has, a brief discussion also in it about some of the species and genus identification parts, as well as, some of the ways you can prepare yourself to go out in your own backyard and study them. So wonderful book as well, I'd like to plug.
Yeah, thanks for highlighting those resources. And maybe we can list some of those in the follow-on email that we'll send out as well, so that people, if they didn't jot it down fast enough, can get it.
Great.
And I'm sure it's something we can get through the library. I request things all the time, and they always find everything. Getting back to some of the life cycle and basic ecology, how long they're in a leaf litter. Somebody was asking just how long is the life cycle?
Oh, wonderful question. So many of the things that I should say, but I didn't say. So I'm glad we have this question and answer opportunity. So we have found that firefly larvae, certainly we know it's going to be a minimum of 1 year, right? Because they're eggs, they hatch, and then they're living as larvae until certainly the next firefly season. But there's evidence that in many cases, it's in fact 2 years that they go through. 1 year, but they're not quite ready to emerge as adults yet. Then they go through another year as larvae before emerging as adults. So I always say 1 to 2 years, because it's also going to be dependent on the habitat they're in, dependent on the species, and certainly also dependent on, up here, it's going to be more likely on the 2-year end of things, because as you go through into the fall and the winter, they just presumably have to go into a completely, sedentary state where they're just overwintering without even feeding, just kind of, the equivalent of hibernating for a while deep down in the soil below the freeze level, or maybe even within the freeze level, they may be able to survive. But that's, of course, different as you get further and further south. They may be able to, be feeding more of the year, and then be able to, go through a life cycle more quickly.
Do you know if, as we head into potentially, drought conditions today aside, does that, that's going to limit the activity of their prey items, and does that slow down? Like, how much will that send a species, maybe, or an individual, into a 3-year cycle, or is there on that?
Yeah, it's a good question. And I don't know the answer to it. We do know, as I mentioned, there's a lot of reason to believe that, they're just not going to survive in a place where it's just too arid. And so it's possible that some species could survive and reemerge in a dry area, again, go into some form of senescence, some form of, like a hibernation, and then be able to emerge. But nobody's documented that. And I'm much more concerned that in many places that are, where it's, just moist enough for a particular species now, if it gets too dry there, that particular population will just be wiped out. I think one of the interesting take-home messages for all of this is to remember that you have adult fireflies for a narrow window of time, right? You have them for maybe 1 to 2 months in a particular habitat. And the other 10 months of the year, the population is entirely dependent on the survival of those larvae. There's no way they're going to be replenished if those larvae don't make it. And unfortunately, so many of our practices that we engage in as humans can potentially negatively impact those larvae. I've mentioned things like, over mowing, pesticides, herbicides. when we think about how we build housing developments and go over and just turn over all of the soil, presumably, as you regrade places, you're just tearing up that thin living layer of soil and generally burying it in a pile deep under lots and lots of unliving soil. And it's very hard to imagine a population of firefly larvae that surviving that kind of traumatic destruction to that soil habitat. So I do want to point to that. I've even had discussions with people when I've gone out to study the New Mexico fireflies, where and again, it's more of an issue places like that, but they've had people comment that they never see fireflies where cattle are grazing. You can imagine another organism besides humans that can have, when being managed by humans in large groups in a using very limited water resources, just trampling all of the habitat along the water that has the appropriate moisture for fireflies, and just completely churning up and destroying that, again, thin layer of biologically active soil where you would see the soft-bodied invertebrates and the fireflies that feed on them. So anyway, that's
it's very sobering to consider how easy it is to get rid of your very local populations then. And I'm wondering, like, how far do they disperse when they're adults? Because they're not going to be dispersing as larvae at all.
Yeah. So the answer to that question is an interesting and challenging one. And actually, yeah, I've even had to call into question the other part of your hypothesis there, that they're not dispersing as much as larvae. There is some reason to believe. There are some fascinating examples studied, especially in Europe, of some rapidly dispersing larvae and some rapidly dispersing firefly populations. Because I always used to tell people until this research came out that it seems very unlikely that they would disperse very far, because just like you, I thought these tiny little larvae living in the soil are not going to disperse far. And I still think that that's true for many species, but certainly there were these wonderful talk by some Scandinavian researchers that had fireflies migrating across firefly larvae migrating across highways and attaining high average speeds. But that's a very different species than we have here. And again, you have to assume that they're going from one appropriate habitat and finding another. It's limited. And then the adults, this is the important point, my research. So if you think about male fireflies, of course, they're flying. They have the potential to disperse. I will say that, my research all shows that they are incredibly responsive to other flashes of light. And so I don't tend to see fireflies wandering off away from a site where there's lots of other fireflies. They tend to circle back in because they're attracted to those flashes of light. Even if they're not a female flash, they're attracted to any flash. The times that I do sometimes follow a male and see him just wandering off and off and off is when things have died down and it's the end of the evening and he doesn't see other flashes of light, and then he just keeps going and going and going until, hoping to see something. But the important point to that is it doesn't matter how far a male goes, because he does not carry the next generation. So it only matters how far the females go. And the fireflies I've studied, and again, this is one species. I know a colleague of mine just, did another species. And so we've seen fireflies where the females are incredibly loyal to their site. In other words, you can catch a female, mark a female, and you can come back to her night after night after night, and she's staying in the same spot. On the other hand, this, not me, but this other researcher did find one female moving like a meter at a time one night after another. So over enough nights, that one would have gotten pretty far. And what's really fascinating is this other research out of Europe where there is a South American firefly that is now an invasive species in Spain and southern France, I guess. And yeah, in the Pyrenees area, it has been spreading by miles and miles from year to year. And I have to have some conversations with those people to get a better understanding of what they think is really going on, because again, it's a very different species, and may may just be its behavior, that it spreads very much. The other thing is, for it even to get to Spain from South America, required some human intervention.
Right.
And the question remains, is human intervention playing a role in its spread within Spain? And that's an open question. So it is, if we talk about fireflies living in soil and leaf litter, are there ever cases where we transplant firefly larvae without knowing we're doing it? So that's another. So a lot to chew on there, I know.
I feel like I could ask you about it all night, but I think we're winding down. I'll just ask you two very different questions before we close out, if you don't mind.
Yes.
One totally different than the other questions was, does the chemistry of firefly luminescence have any commercial applications?
So the answer is yes, although it is essentially a research application, but the research, I think you would argue, has enough commercial value, because it's biomedical research generally, that it actually for many years drove a market of firefly collecting across the United States, where people would actually be paid to collect fireflies in order to get the luciferin and luciferase, the two chemicals responsible for this. And the reason for that is that luciferin and luciferase are a substrate, the chemical that is going to be acted on by the enzyme, and the enzyme luciferase. So luciferin is the substrate, luciferase is the enzyme, and the luciferin, in the presence of luciferase, undergoes a transformation that allows it to release photons of light. Its electrons get excited and then relax and release photons of light. But the key element is that it's not just luciferase that has to be there. There has to be oxygen and ATP, adenosine triphosphate, which is, again, we go back to our middle school, high school, college biology, but that is sort of the universal cellular energy source. When we take in sugars, we have tiny little organelles in our cells called mitochondria that convert those sugars into ATP that we use to power other reactions in our cells. And so for many, it has always been a wonderful assay for whether a cell is biologically active. If the cell is producing ATP, is alive and active, and you put luciferin and luciferase into it, and of course it's an environment where there's oxygen, then that cell will glow under your microscope, or even glow without your microscope. And so people have been using that in a whole host of different ways. Of course, now we no longer have to collect fireflies to do that. Luciferin and luciferase can be produced chemically. And so there's no longer a market for people collecting fireflies, which is good, because that would have been, again, really bad for a number of firefly populations if people were still doing that. So that's kind of its main function in any kind of commercial usage, is for research around other chemicals that might be commercially useful, by where you're doing assays of, is a cell alive, is it not alive, is it producing ATP, so forth.
Thanks, that was pretty interesting. So I guess I'll close with the final question of, in your opinion, what is the best place on Earth for watching fireflies?
Oh, geez, wow. So I'm not going to commit to one place, but because one of the things that we are very concerned about with firefly tourism is that it quite often has a negative impact on the fireflies that we're studying, or not just that we're studying, but that we love and that we think are so important. So my goal is always to get people to appreciate and enjoy the fireflies that they have. And that doesn't mean not to go to as many of these other fantastic places that you can go to see incredible fireflies. But I am very cautious. I mean, I love the fireflies of Southeast Asia that light up synchronously in these mangrove trees. That's beautiful. That's wonderful. But we know that those fireflies are, sometimes bearing the consequences of, people coming in with, more and more motorboats to go along the rivers to see them, in the wakes of those motorboats, then changing the nature of the, of the hydrodynamics of the shorelines there. And we know, there's a wonderful display of, people seem to love the synchrony a lot. There's a wonderful display of synchronous fireflies that I've never seen in Tennessee, in Smoky Mountain National Park. You have to, as soon as the tickets come out every year, you have to reserve your tickets to get a bus ride for one night of firefly observing in Elkmont, Tennessee, to watch these Photinus carolinus fireflies, where in this case, they're not perched in mangrove trees. The males are like our males around here, flying over fields, flashing, but they start to synchronize their their multiple pulse flashes with each other as they fly. I'm sure, I haven't seen it in person myself. I'm sure it's remarkable, but if you're interested in synchrony, there are other synchronous fireflies that you can find in North America. Photuris frontalis is a species that will synchronize its flashes as well. So you just have to, catch them on the right night in the right habitat. So I would say, yeah, please go and see them where you can, when you can, and be cautious with them, be careful with them. And I bet there's going to be other incredible fireflies. We talk about 2,000 plus species because people, like many invertebrates, people are always discovering more. So we'll discover more, and hopefully we'll protect the ones that we have as well. That's my dancing around the question answer.
It was a great answer. I appreciate it. And thank you for answering a ton of questions I've asked you tonight. And I really appreciate, I feel like I could ask you a ton more. We'll get a chance to do that hopefully next Tuesday night when we see you for a walk. For those who are still listening, thanks for hanging in there tonight. And we'll look forward to continuing the conversation then. And I want to pass the baton back to Matt at Cary Library and wrap up for this evening.
Thank you, Chris, and thank you, Sarah. Thank you, everyone, for attending this program. That was recorded and will be on the Cary Library YouTube page in just a few days, and you'll receive an email if you had attended tonight or registered with that link. So thank you again, and have a good evening.